What we have up here then are neurons that are going to be receiving sensory information from a number of different spots of the body and then sending sensory information or sending motor information to the muscles that control it. So if you look on panel A there, we see a neuron on that figure on the left that's sending an axon from one of these spots in the precentral gyrus down ultimately to a muscle as it controls the contraction of that muscle at the motor end plate. Neurons in the left side of the brain are going to be controlling the contraction of muscles on the right side of the body. There are exceptions to this, but primarily it's a left, right, or contralateral control. So we see that neuron sending down an axon, and then as we move through sections of the brain, we can see it going through a portion of the midbrain, going through a portion of the pons, and then entering the medulla. We're at something called the medullary pyramid. There's going to be what's called a decussation or a crossing over of that axon, where that axon will then go over to the contralateral side and then move down the spinal cord in what's called a corticospinal tract, where it will exit the spinal cord and then innervate or synapse with a muscle. Also in panel A, now we see how sensory information is going to make its way up to particular parts of the thalamus and ultimately then parts of the cortex. So here we see a tract, a collection of axons found within the central nervous system, this one in particular a reticulospinal tract, in which we see axons that are moving up the spinal cord through the reticular formation and then decussating at particular points within the brainstem, then traveling up to the thalamus and then to specific parts of the cortex. We can see information there on panel B, for example, in which we see some primary sensory neurons that are going to be carrying information up to the somatic sensory area of the cerebral cortex, unlike what we just saw on panel A with motor sorts of sensory information. This is our primary sensory pathway, in which that decussation is not going to happen up at the level of the midbrain, but that decussation will happen primarily within levels of the spinal cord and medulla oblongata. Before then, those axons make their way up what's called the mediolemniscal pathway, up to the thalamus, and then to the somatic sensory area of the cerebral cortex. A little bit about the spinal cord here, and we'll talk about this in a little more detail when we discuss in the next lecture things like multiple sclerosis and amyotrophic lateral sclerosis, for example. Here we see the spinal cord and the number of levels of the spinal cord from C1 all the way down to the coccyx, for example. The spinal cord is going to be within a foramen found within a vertebrae. As we can see there, there's the body of the vertebrae and the opening within it that allows the passage of the spinal cord. It's only going to be about the width of one's index finger, for example. Spinal cord consists of white matter and gray matter, of course, just like other parts of the central nervous system. Notice the anterior body of the vertebrae there, and the spinal cord in the opening, again, that's posterior to it. We see it perhaps layers of the meninges that we'll talk about in a moment. There are fibers of passage that are going into the spinal cord through the posterior root, and then fibers that are coming out through the anterior root, sometimes more commonly referred to as the dorsal root and the ventral root, and those axons converge to form ultimately these individual spinal nerves, of which, again, there are 31 pair. There's a spinal ganglion associated with this particular structure. That spinal ganglion is going to contain cell bodies whose processes then enter into the spinal cord through that posterior or dorsal. In addition, we can see some of those sympathetic ganglia that mentioned before, the paravertebral ganglia that receive information from the preganglionic cell fibers of the sympathetic nervous system. In addition, we can see gray matter within the spinal cord in sort of a butterfly or moth shape within it, and a central canal that runs right down the middle. The spinal cord contains a number of different tracts, and these tracts are going to be carrying either sensory information, as indicated in blue here, or motor information, as indicated in red. The motor information, we can see one of those particular tracts known as lateral corticospinal, as mentioned before, a cell from, say, the primary motor cortex and the precentral gyrus of the frontal lobe, making its way down from the cortex through the spinal cord, out via a spinal nerve, before that its anterior root, and making its way then to some target skeletal muscle. Here we see some sensory pathways that are going to be carrying axons up to ultimately, say, the thalamus, then off to the cortex for more specific processing. The two primary functions of the spinal cord are going to be in relay and reflex. The relay is indicated by this particular figure here, which we see fibers of passage that are making their way up and down the spinal cord, carrying in sensory and motor information. The reflex arc of the spinal cord is one thing to indicate the other primary function, which of course is in reflex. The reflex is going to be an involuntary response to some particular stimulus, like, for example, that seen by the patellar tendon reflex, which is going to cause extension of the leg at the knee via contraction of the quadriceps muscle, which would be the effector in this particular system. So the striking of the patellar tendon with, say, a reflex hammer is going to activate a stretch receptor that will be carried by a sensory neuron, ultimately to the dorsal root ganglion. It's information traveling through the axon at that particular level, and then in through the posterior dorsal root of the spinal cord. Notice that those cells in the dorsal root ganglion have only one process coming off them. They are, by definition, unipolar neurons, and the one process comes off them and splits, and almost the entirety of that process is going to be an axon, save for a specialized dendritic ending, which in this particular case is the stretch receptor. Cell bodies of these sensory neurons lie in the dorsal root ganglion. They will be carrying information into the spinal cord, where they may or may not synapse with an interneuron. The vast majority, well over 99% of neurons within the nervous system, are known as interneurons. By definition, neurons that are between sensory neurons and motor neurons. You can think of interneurons, or associational neurons, as your book refers to them in some spots, as essentially making this decision about whether or not to activate a motor neuron based on the amount of frequency of sensory information that's passing to it. This is an example of a couple of different types of synapses. You can have a monosynaptic synapse, in which there's interaction between the two neurons, for example, and therefore more than one synapse. Remember that the vast majority of neurons have these multisynaptic inputs from a number of other types of cells. So here we see sensory information coming in via that blue pathway, making its way to a motor neuron, an interneuron, which can make a decision about whether or not to activate that motor neuron and therefore cause contraction of the quadriceps muscle, the effector. This is a protective reflex, and of course is a reflex to determine whether or not there's damage in that particular pathway, especially at a particular level of the spinal cord. So the two functions of the spinal cord, relay and reflex, also gives a very good example of the functional classification of neurons. And neurons are either sensory, they're motor, or they're interneurons or associational neurons. Sensory neurons, by definition, are going to transmit impulses from sensory receptors to the central nervous system. Associational neurons are going to carry information ultimately from sensory neurons to motor neurons, so as indicated here, from neuron to neuron. And then motor neurons will transmit impulses from the central nervous system to an effector organ. Here, of course, we're talking about either skeletal muscle, smooth muscle, or cardiac muscle. Neurons are carrying bioelectrical information, and therefore there has to be a way to insulate the signals that these particular axons are carrying, and that's made possible by a lipoprotein substance known as myelin. Neurons do not make their own myelin. That myelin is actually made by glial cells or neuroglial cells that are found in nervous tissue. Neuroglial, literally translated, means nerve. And these are supporting cells of the nervous system that do have a variety of specific functions, but in general are going to do just that. They're going to allow neurons to do their job by, say in this case, insulating a particular axon. The myelin that insulates the axons of a neuron in the peripheral nervous system is known as a Schwann cell. These individual Schwann cells will wrap around an axon, be filled up with this lipoprotein material, this myelin, and allow then for the insulation of the axon that way. The thing about the Schwann cells, though, and the way that myelin insulates an axon, is that the myelin does not wrap the entirety of the axon. There are actually small spaces between the myelin sheet that makes its way, that is present around some particular axon. These spaces are known as nodes of Ranvier, as indicated regular interruptions of the myelin sheet. Earlier in the semester we talked about how, for example, excitable cells like neurons make action potentials, and then ultimately what the purpose of the action potentials are. We mentioned at that point that if a muscle cell produces an action potential, what it's going to do is it's going to contract or shorten. When a neuron produces an action potential, what it is going to do is release a neurotransmitter by exocytosis. This action potential is going to begin in the membrane that's associated with the soma or cell body of the cell, and ultimately, at a point where the axon leaves the cell body, a funnel-shaped region known as the axon hillock. It's the job of the neuron to then maintain that particular strength of signal all the way down the end of the axon, even though some of these particular neurons can be, say, a meter or more in length, if we're talking about some particular axon that's exiting from the spinal cord and then making contact with some particular target muscle, say, near the foot. So how, given that there's resistance, given that there will be the loss of current and voltage ultimately as one moves along the length of an axon, just like there is an electrical wire, there are these regular interruptions of the myelin sheath known as nodes of Ranvier that have at them a very high concentration of voltage-gated sodium channels. Think of these as little batteries that are wired into the axon along the length of it, and their primary function is to keep the strength of the signal the same as it moves down the end of the axon. So what technically, as an action potential propagates or moves down an axon, what it's doing, it's undergoing something called saltatory induction. So the flow of ions between segments of myelin rather than along the entire length of the axon. Most of the axon is going to be insulated, save for particular spots, nodes of Ranvier, that aren't, that allow for the influx, that allow for an inward movement of sodium to make sure that the strength of the action potential remains the same along the length of the axon. Saltatory conduction, that term saltatory comes from the Latin word saltare, which means to leap, and it's often described that action potentials, quote, leap the length of the axon. What the axon can do, even though a neuron only has one of them, is it can diverge. So axons can branch to influence many neurons. How many? It could be thousands. And an axon can branch, forming these collaterals, and therefore making contact with a number of different neurons. What a particular neuron that's receiving a signal from an axon can have is a case of convergence, in which you have branches of numerous neurons converging on one or a few neurons. Neurons receive signals via dendrites, and let's say that one particular neuron is receiving signals from 100 different axons. Those would be convergent signals. Then that neuron that's receiving those signals branch, and the signals ultimately diverge. And this process allows for some complexity, of course, of neuronal signaling within the nervous system.